Welcome back. Today we're going to talk about meiosis, which is the other type of cell division we mentioned a little bit in the last notes video. First, I strongly recommend you watch the Amoeba Sisters video about meiosis because they will explain the same things, but they've got nice animations that go along with it. Especially if you've never seen anything about meiosis before, the stuff I'm going to go through now is going to make a whole lot more sense if you watch that video first, and you'll be hearing it for the second time, so it'll really sink in for you. It's definitely worth the extra 6 or 7 minutes of your time to watch that one, and then come back and pick up where you left off here. You can even watch it with your cat if you like. So this shows mitosis and meiosis side by side. The biggest difference you'll notice is meiosis produces 4 cells. So there's actually 2 division events that happen. One cell splits into 2, and then each of those cells splits into 2 again, giving 4 cells. But if you look at the numbers that you see, in mitosis, you end up with an exact copy of the cell that you started with. So 46 chromosomes, and both cells have 46 chromosomes. In meiosis, you have 4 cells, and they have half of that, or 23 chromosomes. So we're making cells that have half as much DNA, but also in mitosis, you get cells that are identical. In meiosis, you get cells that are different from each other and different from the cell that you started with. So now we got to go through all the steps that explain how we get to that point. So with meiosis, this is done by eukaryotic cells, and these are germ cells. So they're the cells that are going to divide to become eggs and sperm. You start with one cell that is diploid, or 2N. We'll talk more about diploid and haploid. That just means it has 2 copies of each chromosome. When you're done, you have 4 gametes. Gametes are another term for eggs or sperm, and each of those are haploid, meaning they only have 1 set of each chromosome. Or one copy of each chromosome, and those gametes at the end are genetically different from each other, and they're different from the cell that you started with. Some important vocab that you'll see throughout this presentation. First is the idea of sexual reproduction. So that happens when an egg and a sperm combine to make offspring, and that's only possible if the egg and the sperm have a half set of DNA. So when they combine, half of the DNA from mom plus half the DNA from dad combines to give you 1 full set of DNA in the offspring. That's why offspring are different and similar to both parents, because they don't have the exact same DNA as mom, or they don't have the exact same DNA as dad, but they have some DNA from both. Asexual reproduction is not meiosis, and that's mitosis or binary fission, because that gives you an exact copy. Homologous chromosome is the term we use for the 2 copies of each chromosome that you get from each parent, or the one that you get from each parent. So in the human body, we have 23 types of chromosomes, but you get 1 full set from mom and 1 full set from dad, meaning you have 46 total, but you have 2 copies of each one. Each type of chromosome is called a homologous chromosome. Ploidy, kind of a weird word, is a measurement of the total number of sets of DNA. Today, we're only going to worry about diploid and haploid organisms, but there are other species where there's triploid or tetraploid. Some plants, I think, can even be more than that. But for now, diploid or 2N means you have 2 copies of each chromosome. Why do you have 2? 1 set from mom, 1 set from dad. Haploid is represented by the letter N, and it means you have just 1 set of chromosomes. Here is a karyotype. That's simply just a picture of someone's chromosomes. And you can see, if you wanted to count all those, there would be 46 chromosomes total, but clearly they come in pairs. You have 2 copies of each one because you got 1 copy from each parent. So this is a diploid karyotype for a person. Next vocab word is a tetrad. A tetrad is a pair of 2 homologous chromosomes. So you can see a tetrad here. When we're going to see them, tetrad obviously has that root tetra, which means 4 in it. And you can see 4 total chromatids. So these are sister chromatids that are bound at a centromere. So we count this as one chromosome. And these 2 sister chromatids are bound at the centromere. So we call this one chromosome. So we'd have 2 copies of the same chromosome, one from mom, one from dad. When they're paired up together like this, we'll see that in prophase 1, they form a tetrad. Another thing that happens with tetrads is crossing over. So you can see when you put these 2 chromosomes next to each other, because they're the same type of chromosome, sometimes the ends will sort of flip-flop with each other. And you create these hybrid chromosomes that have a little bit of information from mom and a little bit of information from dad. That happens during prophase 1 and is an important thing that increases the genetic differences in the cells that we get from meiosis. Next is the idea of independent assortment. This is something you'll see during metaphase 1. But when the chromosomes are lining up in this step, the arrangement is random. We'll talk more about this a little bit later on, but you can see here 2 different arrangements of chromosomes, and it's just random. This one happened to be with the blues on the left, reds on the right. This one happened to be blue, red, red, blue. And without going through all the steps, you can see that down here, you end up with different cells than you got over here. The reason we have to worry about this now in meiosis, we didn't have to worry about it in mitosis, is the chromosomes are lining up next to each other, side by side. If you go back and you look at the notes for mitosis, you see they lined up single file. Now that we have them double file, there are different ways they can arrange, which can result in different daughter cells at the end. So, like I said, meiosis has 2 divisions. The first division we call meiosis 1. Cool, that makes sense. We start with a single cell, a cell that is diploid. When we are done with this, we're going to have 2 cells, and they're both going to be haploid. That's why this is called a reduction division, because we cut the number of chromosomes in half. There's a link to a simulation down here. Super helpful. I think it's really a great way to visualize this process. I'll show it to you at the end of these notes. So we start with interphase. That's pretty much the same as it was for mitosis. The cell is going through G1, S, and G2. Does its normal function. DNA is unwound in chromatin. That's like the spaghetti format. The DNA gets copied, making 2 copies of each chromosome. And the cell is diploid at this point because there are 2 copies of each chromosome, one that you got from mom, one that you got from dad. Next comes prophase 1. In this step, the DNA forms tetrads. So homologous chromosomes pair up. So the copy of chromosome 1 from mom pairs with a copy of chromosome 1 from dad. Chromosome 2 from mom pairs with chromosome 2 from dad. Crossing over can happen at this stage, prophase 1, where the homologous chromosomes will switch little pieces. Centrioles move to the poles, the opposite ends of the cell, and the spindle fibers will start to assemble. The cell is still diploid here. You can tell that because if you look at our cell, we've got 4 chromosomes total, 1, 2, 3, 4. We know it's diploid because there are 2 copies of each, 2 big, 2 small. Next comes metaphase 1. The DNA is in tetrads. Spindle fibers attached to the centromeres, and they line up at the metaphase plate. We still have 2 copies of each cell, or two copies of each chromosome, which means the cell is diploid. But here's the most important thing about metaphase 1. Notice how the chromosomes line up double file. This is what really sets meiosis different from mitosis. In mitosis, they all lined up single file, straight down the middle of the cell. But now they're lining up double file next to each other. That's going to make all the difference once they split. Anaphase 1 is probably exactly what you'd expect. So they get pulled apart. You can see them getting separated. This cell technically still has 2 copies of each chromosome, 2 big, 2 small. So it is still diploid for now. That is about to change. So when we finish telophase 1 in cytokinesis, now DNA is still Paired up, homologous chromosomes together, we have two cells, but each cell is now haploid. So how do we know it's haploid? Well, we have two chromosomes here, but we only have one copy of each, one big and one small, one big and one small. So because we only have one copy, now our two cells are haploid. Then we go through another division. The second division follows those same steps again, prophase, metaphase, anaphase, telophase. But now we slap a 2 behind it because we're doing it all for the second time. Remember, we are starting with the two cells we just had after meiosis 1. They're both haploid because they both have one copy of each chromosome. And we are going to end up with 4 cells, and they're still going to be haploid. This one, even though it starts a little different, the steps will look similar to mitosis. So here's prophase 2. Sister chromatids stay paired up. Centrioles move to opposite ends of the cell. The cell is haploid. Each cell is haploid because it has one copy of each chromosome. We're back to metaphase 2, where they line up single file down the middle of the cell. Spindle fibers attach at the centromeres, and we are still haploid. Here, anaphase 2, the sister chromatids separate and get pulled apart for anaphase. If you wanted to be technical here, you might technically say that during anaphase 2, the cells could be considered diploid. The reason is, if you look at this cell, because these sister chromatids have separated, we have 2 copies of the little chromosome, 2 copies of the big, 2 copies of the little, 2 copies of the big. So I know the slide says haploid. You could consider it haploid. You could consider it diploid if you wanted to be technical. I would not ask you, is this cell haploid or diploid, because it's kind of a gray area. And then we get telophase 2 and cytokinesis. So then we split each of those cells. Now they are definitely haploid because each cell has one copy of each chromosome, one big, one small. Nuclear membranes come back. We have 4 cells. And notice that even though they're all haploid, those cells are different from each other, and they're different from the cell that we started with. So in meiosis, we end up with 4 cells that are all completely different. I think that is the end of the actual notes. So there's another Amoeba Sisters video about counting chromosomes. I strongly recommend that one if you're like, wait, chromatids or chromosomes or centromeres, what the heck is happening? That video is going to make all of that clear for you. If you want, you can make a summary of this in your notes. If you have any questions, watch the videos, check the notes, send me a message, show up to office hours. Well, I guess office hours were probably earlier this week. Well, depending on when you're watching this, it's still possible. Let me know if you have questions. I'm here to help.